Thank you everyone for joining today for the E3SM BGC webinar. Our speaker is Dan Ricciuto from Oak Ridge National Lab.
Thanks, Kate. Yep, and I'll be talking today about uncertainty quantification that we've done primarily in the land model, ELM as part of E3SM. I'd like to acknowledge Kachik, my co-author. He's really the driving force behind a lot of this work, as well as Cosman and Vishigan from Sandia, also Dan Liu and Peter Thornton from Oak Ridge. And I'll talk a little bit about some uncertainty work we've done with ELM-FATES. So thanks to Jennifer Holm for helping with that, and also in the crop model as well. So thanks to Beth Juniak for helping out with that work. This work was funded by E3SM and also from our SIDAC project. And a lot of the tools and capabilities were also developed by the Fast Math Institute. So I'll jump in to, oh, there it goes. Some overview and motivation for this work. And so when we look at uncertainty in these global coupled models, a lot of that is analyzed from looking at multi-model ensembles, where we have different modeling groups contributing, and in some cases, few ensemble members contributed by individual groups. And what we often see are large spreads in outputs, for example, cumulative land flux from past to future through 2100. But there are many quantities of interest that these models predict. In addition to carbon flux, we have things like biomass, burned area, leaf area index, hydrologic fluxes. And we don't see much in the way of formal uncertainty quantification from these models, primarily because they're so expensive to evaluate. And in a lot of cases, they have a very high number of uncertain parameters that could be considered. So we call that high dimensionality. So some of the challenges that we have in E3SM and the SIDAC are trying to determine what processes are driving uncertainties and what accounts for key differences among the models. And ultimately, what we want to do is develop the capability to perform calibration so we can bring these data sets together to improve model predictions, historical predictions, which will hopefully lead to improved future predictions as well. So specifically, since most of our work has been in the ELM model, ELM is in itself a complex model with many processes that are considered. So things like surface energy, hydrology, biogeochemistry, and land use changes are all considered. So we kind of inherited this model as coming from CLM 4.5, but since then the model's become even more complex and many things are being considered in terms of new processes. We're thinking about coastal processes. We're thinking more about human interactions, vegetation dynamics, for example. And so these models are becoming more complex over time. But if we want to do uncertainty quantification, generally we need large ensembles. We need the ability to run the model many times to explore parametric and structural uncertainties. For considering BGC, one consideration is spin up. And so typically we have to equilibrate the carbon and nutrient pools with pre-industrial conditions and then move forward with transient forcings. And so that generally requires hundreds of years of simulation and can limit the number of ensemble members that we can run, even when considering land as one of the less expensive components of E3SM. So one way to kind of get around that is development of surrogate models. So surrogate, I'll talk a little bit more about what I mean. Basically a model of the model that's much faster to evaluate, that can increase the efficiency of uncertainty quantification techniques like sensitivity analysis and model calibration. So we've done some work to create infrastructure within ELM to enable more efficient uncertainty quantification. A lot of that work originally focused on single column or single grid cell applications of ELM. And so we've done that, for example, in version 1 with a number of FluxNet sites. We're doing that now with ELM-FATES. And so we're using MPI for Python as part of an offline land model testbed that we've developed called OLMT. And so this basically will take a case and a run directory and clone that many times and manage running lots of ensemble members in parallel, and then post-processing the model outputs into things that we care about and the quantities of interest that we're specifically analyzing for any given study. So when we're doing those kinds of single grid cell simulations, typically we're running anywhere from hundreds to thousands of ensemble members. More recently, we've begun developing infrastructure to enable global scale uncertainty quantification. So most of what I'll present today was actually based on the satellite phenology version of the model, but we're in the process of extending that to a BGC as well. So it's a modified version of the offline land model testbed where we're managing global runs, typically at low resolution, so 2 degree in this case. So I'll be talking specifically about a study where we modified 10 ELM parameters, primarily dealing with photosynthesis and leaf properties. And we ran 200 model simulations and analyzed gross primary productivity, or GPP, as our quantity of interest. So a little bit more information about the parameters that we looked at. So 10 parameters again. So fraction of leaf nitrogen and rubisco, or FLNR, as a strong control on a photosynthetic uptake. So proportional to or directly correlated with VC max. And so we've picked some ranges of that parameter that are consistent with the literature. So you have min and max values there, and then the default range on the last column that indicates what variation we have in the default model across the different plant functional types that we're using. The next 2 parameters, MBB-opt and BBB-opt, are the stomatal slope, stomatal conductance parameters dealing with slope and intercept. The 4th parameter is a rooting depth distribution parameter, so it controls to what fraction of roots are near the surface as opposed to distributed more at depth. The next 2 parameters deal with temperature sensitivities of VC max, and so the first one being an activation energy, the second one being an entropy that controls where basically the temperature optimum appears. The day length scaling parameter affects to the degree that day length affects photosynthesis in the model. So that's a function as the day length becomes relatively shorter, photosynthesis is scaled back. And the last 2 parameters deal with leaf physical properties, dimensions, and orientation. So we took those 10 parameters and those min and max ranges, and we constructed 200 random sets of those 10 parameters and ran them forward in time. And so what we're going to be looking at, the next slide here, is basically just the ensemble mean. And I'm not sure how well this video is going to play over the connection here, but what we're seeing over a 15-year period, the monthly values of gross primary productivity and how they evolve over time. So you can see, hopefully, the seasonal cycles as the Northern Hemisphere becomes productive in the summer, and then less productive or no productivity at all in the winter, with the opposite pattern in parts of the Southern Hemisphere. And in the tropics, some back and forth kind of between high and low productivity, driven by, in a lot of cases, wet, dry season kinds of dynamics, and other areas where the GPP is relatively constant through the whole year, for example, in Indonesia. On the next slide, so something we can do, we can look at the standard deviation over the ensemble. So we have, again, 200 ensemble members that we're looking at, and so this indicates where basically the highest uncertainties are given the parameter ranges that we have used to construct this ensemble. And you can see it has a pretty similar pattern to the mean values that I just plotted. So the uncertainty or standard deviation is strongly correlated with the mean values. In a lot of cases, it's proportional. You do see some areas where perhaps the standard deviation is lower than you might expect given the magnitude, for example, in the Western Amazon. So what can we do with this? What can we do with this ensemble? So one of the first things that we like to do in uncertainty quantification is conducting global sensitivity analysis, or GSA. And so that can tell us which parameters are really driving the uncertainties in model outputs, also known as snowball indices or variance-based decomposition. Again, it's for attribution of output variance to the input parameters. And so it can give us an idea, for example, of how much output variance would go down if we just took a parameter and fixed it at its nominal value. And we can also use it to explore joint sensitivity. So in some cases, a particular output might be sensitive to a combination of two or more parameters. So we did that for this global, at first for this globally average GPP. And so we can look at the 10 parameters that we varied and the relative kinds of sensitivities that we're seeing. And what jumps out, of course, is that this FLNR parameter, the top one here, is by far the most sensitive, which isn't all that surprising since that's a direct control on the VC max or photosynthetic uptake. But we also see other parameters that are identified as sensitive, the model conductance slope in the Ball-Berry equations. Some of these temperature sensitivities are showing up as well. And then on the right-hand side, this just gives us an idea basically of the relative magnitudes of these sensitivities, and also where these bubbles overlap gives us an estimate of where the joint sensitivities are important. So for example, GPP is sensitive not just to FLNR and this VC max SE, but also the combination of those two parameters. So uncertainty propagation can be enabled by surrogate models, which I talked a little bit about already. And so basically we take some set of full model predictions, for example, these 200 ensemble members from ELM, and we develop a response model for those ensemble members. And so in this case, for example, if we're interested in global gross primary productivity, we know the values of that for the 200 ensemble members, and then we can fit a function over the whole parameter space that will predict what that value of global GPP will be at some other combination of parameters without having to run the full ELM model. So we work with the model as kind of a black box. It's not in a non-intrusive method. We don't need to edit any of the ELM code to do this. We just run some ensemble and use that to build these surrogate models. And so you may have heard other names for these types of models: proxy or meta model, emulator, or response surface models. So I'll talk specifically about a method that we use for looking at this global scale surrogate model. So this is a method that Kachik developed. It's a Spatio-temporal surrogate model via Karhunen-Loève expansions. And so a big problem with these global ensemble members is we have a huge number of outputs. And so you can consider each value of GPP over every grid cell and every time point as a separate quantity of interest that we need to estimate with a surrogate model. So in the 2-degree offline model, we have over 3,000 active land cells, and in this case, we're looking at 180 months of output from 2000 to 2014. And so that's over 500,000 values or outputs for GPP. And so what we're doing with this method is to reduce the dimensionality. And when we look at those output fields, if you remember what those videos look like, you can imagine there's very strong spatio-temporal correlations that exist within that model output. And so the GPP is, the variations in GPP are similar to the variations in the grid cell next to it. And they're similar to, a year prior or a year in the future. So think of principal component analyses in stochastic space. So we take those 500,000 outputs. Instead of developing surrogates for all of them, which would be very time consuming, we only build about 2,000 of them, one for each eigencomponent. So the end result is a single surrogate that is resolved in space and time and has a relatively low relative error compared with the true ELM. And so we can test the accuracy of this surrogate using some validation points and seeing how well the model or the surrogate model matches ELM predictions at a point in the parameter space that we didn't use to train the surrogate model. And so once we have the surrogate model, it is extremely cheap to evaluate and can be used for various purposes, for example, to conduct these sensitivity analyses or eventually to calibrate model parameters with data sets. So the surrogate model accuracy, this shows the relative error of the surrogate model as averaged over all the land grid cells. And so we can see kind of a seasonal pattern to these relative errors and where these peaks occur. I believe that's the month of August in most cases. And that's when we see kind of the most complex behavior of GPP. GPP is influenced in these late summer months, and so since we're considering, a lot of the land grid cells are in the northern hemisphere, and what's happening is by the late summer, we start to have water play a bigger role in the predicting GPP through either VPD, vapor pressure deficit, or soil moisture, and so the response becomes a little bit more complex than it does, say, in the early spring, where it's really driven by these FLNR, VC max values. So we can take that surrogate model then, and we can begin to do some analysis on that. And so one of the first things that we looked at were some single grid cell level sensitivities, and these are 2 tropical sites. And so we're looking here at the top at a site in Brazil. Each bar plot represents a month, and so we have 180 months in total that we're looking at. So we can see the relative influences of different parameters and how they vary over time. And so basically the height of the bar represents its the sensitivity of that parameter, how much output variance that parameter is controlling. So in a lot of cases, it's the FLNR that's the largest single driver of that variance. But we can see kind of a pattern. In some months, the MBB opt, or stomatal conductance slope, becomes more sensitive. So the relative height of that green bar is increasing, and that in a lot of cases corresponds with when conditions are somewhat drier. And the same is true with the stomatal conductance interceptor, this BBB opt parameter. We also see for this Brazil site the VC max SE, or entropy parameter, playing some role in sensitivity as well. And so where that temperature optimum is in the calculation of VC max is important at this site. Whereas on the bottom site, it's a Botswana. It's a little bit, it's quite a bit drier in terms of, it's more kind of a shrubby or savanna kind of site. And so we see a relatively larger influence of the green, the stomatal conductance slope parameter, relative to FLNR. And we also see this yellow one, the routing depth distribution parameter, showing up as quite a bit more sensitive, indicating a greater role of soil moisture in driving the magnitude of photosynthesis, or GPP, at the site. The next one, I'm show a couple of temperate sites. So the top one being Harvard Forest in Massachusetts. And so that's a relatively wet site, and we see the FLNR really driving the sensitivities or the variances in GPP at that site. Although we do see these stomatal slope parameters becoming important in the growing season months, and then in the non-growing season months, the day length parameter and this VC max entropy parameter becoming more important. Although during those periods, the magnitudes of GPP are quite small. And the bottom site is, I believe, in Southern California, again, a much drier site, so we can see the strong role of both the somatal conductance slope and intercept parameters there. So with this globally relevant surrogate, we don't have to look just at individual grid cells. We can begin to look at global distributions and global patterns of sensitivity. And so I'll show a few of these. And so this is the sensitivity of FLNR, or the fraction of leaf nitrogen in Rubisco. And this is month by month. So I'll play this video and it'll go from January to December. So you can see how over time, the spatial distribution of these parameter sensitivities are changing. And so for FLNR, it's quite important in most places, most of the time, that you can see in the winter months in the Northern Hemisphere where not much is going on, that parameter becomes less sensitive, and things like day length and temperature sensitivity are becoming more important. So I think the next one is a little bit more interesting, where we look at the somatal conductance slope parameter. So this is MBB opt. So you can see in January, we see some sensitivities in Canada and Siberia, and then in Africa and the Sahel region as well. And so as that advances over time, you can see sensitivities increase and then decrease in the Sahel region following wet and dry seasons there. And you can also see April, May, June sort of transitioning from less sensitive to more sensitive in different parts of the Northern Hemisphere in the late growing season, July and August. We start to see this parameter becoming quite a bit more sensitive in the northwestern US and parts of Canada. You can see some patterns in Asia and in eastern Brazil as well. So this tells us basically when and where specific parameters are more important in driving variations in GPP. So it can give us some clue as to when we might need to measure or understand these parameter values better. The next one is the VC max SE, so the temperature sensitivity, controlling the temperature optimum of VC max. And so you can see that parameter quite important in kind of the early season in the Northern Hemisphere. Basically, it's kind of controlling when photosynthesis may begin in terms of where this temperature optimum is, but also some sensitivity in Brazil and in other tropical regions as well. So possibly indicating where, in some cases, for some values of this parameter, areas might be getting too hot for optimal photosynthesis. And then the last one here being the rooting depth distribution parameter. And so we can kind of see similar patterns that we saw with the somatal conductance slope, the Sahel becoming important at certain times of the year, and in general, indicating when soil moisture may be a limiting factor for photosynthesis. So we don't just have model outputs. It's for GPP, the observation that we're interested in. We have gridded observations and we have benchmarks, and so it's something that exists in ELM already that we've spent quite a bit of time looking at in these BGC experiments. And so the nice thing about this, we can play the video here and look at the seasonal and interannual patterns of GPP as well, and it's quite similar to what we saw when we looked at the model ensemble mean. And so this variable is directly comparable with the model GPP output, and it can be used for calibration of model parameters from single grid cells to kind of global scales. And the methods that we're developing here will be extensible we hope to other ELM products or other gridded types of observations that we might want to use to try to constrain model predictions in the future. And so a little bit more about inverse modeling. So we want to eventually tune or calibrate model parameters with observational data. Generally, that requires, again, quite large ensembles. And so that's something where the surrogate models can help us that are fast to evaluate rather than having to run many model simulations. They can also incorporate observational error or noise. And so that gives us, given that, some idea of what the optimal parameters in the model are and what the uncertainty around those parameters might be. And so for these kinds of problems, inverse problems, Bayesian approach is the main tool that we're using for things like calibration. So Bayesian inference allows incorporation of various sources of uncertainty. A method that's quite popular is Markov chain Monte Carlo, or MCMC, that we can use to build posterior probability density functions. So nonlinear models like ELM, generally often they will lead to, when we do calibration, to ugly kinds of high dimensional parameter PDFs. But there are advanced techniques available to deal with that. But they do require many online, mostly serial observations of the model. So something like MCMC, being kind of a chain, is hard to parallelize. We can run multiple chains to make things more efficient, but in the end, the algorithm is not easy to parallelize. And so this is why surrogate models are quite handy, because we can run a set of model simulations, an ensemble, and then develop a surrogate method, surrogate model based on those, and those are very fast to evaluate, even if we must do it in serial. Predictive uncertainty so when we do these, when we construct these surrogates, of course, we're introducing an additional source of error to the problem. The surrogate isn't going to be 100% accurate in every case, and so there's additional errors that come with the surrogate model. But we can estimate that as part of this process, how much error the surrogate is contributing, and also observational noise as well. And so some initial work towards that. So we've taken this globally relevant surrogate model, and we just pulled out one grid cell centered on the US Missouri Ozark flux site, but this isn't actually using the data from the site. It's using the globally gridded GPP product and the model output from that grid cell. And so what we can see is the prior kind of spread of GPP is quite large. And so that's from the 200 parameter sets that we used in the original ensemble. And then when we apply the calibration using the data, which is represented by the black points here, we can estimate the posterior prediction uncertainty for GPP. And we can see that by the green shading is a much more constrained prediction than we had from the prior. So the first point is the calibrated model is able to reproduce the data points with high accuracy. Posterior uncertainties are greatly reduced. And what we can do then, since we have this, we can apply this surrogate model anywhere. We want to try calibrating many grid cells, and we can do that either for all 3,000 grid cells, or we can aggregate by region or by plant functional type to try to get posterior parameter predictions, probability density functions that are regionally relevant. But I think one interesting point will be doing this grid cell by grid cell to look at the spatial distributions of these posterior parameters. We can also begin to look at embedding model structural error. And so these methods kind of assume implicitly that we have a perfect model, and the only potential problem is the parameter uncertainty, right? But we don't always have that, and there may be structural errors that are contributing, in which case we can't simply just calibrate parameters. But we can deal with structural error by embedding it into kind of our assumptions about the parameter errors themselves based on model physics, and use that to do meaningful extrapolations to full sets of predictions of our quantities of interest. And so we've begun to look at that also for some of the FlexNet sites to try to estimate potentially how much model structural error is contributing, and compared to things like surrogate error or posterior uncertainty. And so we can see at this particular site, model error represented by the lighter blue color is responsible for a lot of the prediction uncertainty, more so than the posterior uncertainty in the parameters or the surrogate model. So we're also looking at how we can improve surrogate models further. So I think these dimension reduction techniques can go a long way at these global scales. But we do note that in a lot of cases we're dealing with output time series, and so machine learning techniques that are able to handle these and to look at the properties of these time series. For example, the LSTM method, or long short-term memory, is a type of neural network that can use the past predictions to inform future predictions, and we can build into that some of the known physical connections in the model to make that more efficient. And so this particular work, we started with a simplified version of ELM, where it's just looking at a few of the processes within the carbon cycle allocation, for example, phenology and decomposition. And we built this physics-informed LSTM model to account for some of the connections between the quantities of interest that the model predicts. For example, GPP, NPP, net primary productivity, NEE, net ecosystem exchange. And so when we do this, we have a physics-informed model. We find that it can be much more accurate than an LSTM that makes no assumptions about connections or physics within the model. In this case, at a specific site that we were interested in. And so the goal here is to reproduce the ELM model. We're looking at the right-hand figure, the ELM represented by the dashed blue line predicting GPP. And it's compared with 3 methods of building surrogate models. So MLP, or basically a neural network that's not taking into account some of the time series information, compared with a LSTM model, and then also compared with the tree or physics-informed LSTM model, where we're building these surrogates based on 47 perturbations and 47 model parameters. So a pretty high dimensional problem in this case. So you can see the red line, or the tree LSTM, is much more accurately predicting the original ELM GPP. So I think there's promise that we can build on these methods, combine them when we can, to create even more accurate surrogate models than we already have for these global problems. So I'll also talk briefly a little bit about some of the work that we're doing with ELM-FATES, and so that's quite a bit more complex than the plain vanilla ELM that we've been working with in the past, and so it contains a lot more parameters. And so we're looking at doing sensitivity analyses at a boreal site in Alaska, and looking at how these parameter sensitivities vary as a function of time. So this particular example is leaf area index. And so in this particular case, we started with on the order of, I think, 20 parameters, and found that really a lot of the variations are driven by only 4 of them. And so that kind of parameter dimension reduction is important for eventual calibration. More recently, we've been looking at this site in more detail for a number of additional quantities of interest and for multiple plant functional types. And so in this case, we're looking at 3 plant functional types that are known to grow at this site. We've also turned on the FATES hydro model. And so when we do these things, we have a lot more uncertain parameters than we considered before, so nearly 200 parameters. And so with such a high dimensional space, there is some concern that we need a very large number of model simulations to try to make any sense out of what the sensitive parameters are and to do eventual calibration. But we are starting to see some things that are making sense. So the hope is that we can take those 200 parameters and identify the most sensitive 15 or 20 of those parameters in order to do a calibration exercise and get the model performing better at that site. We're doing a similar exercise with the ELM crop model. Again, more complex than the original ELM, containing additional parameters that have to deal with things like grain and crop phenology. So I think in this case we started with 20 parameters and looking at a soybean site, but we are starting to see things that make sense there as well, things that control GPP and MPP, so things like specific leaf area at the top of the canopy, the carbon-nitrogen ratios of various plant components. And we see things that are kind of logical too, as in the root carbon is driven by root parameters. And we see an output variable like net ecosystem exchange, which is very integrative. So it's the sum of many different processes that are happening in the model. So respiration, photosynthesis, and so we see often when we're looking at any E that since it has more sensitive parameters driving it than some of the other variables, and so I think that's consistent with the level of complexity that goes into calculating net ecosystem exchange. So when we're dealing with these very complex models and very high dimensional problems, one thing that's coming to mind is the ability to kind of break the model up and examine different pieces of the model for specific quantities of interest that might be relevant. And so we've been looking at developing functional units that take pieces of or process submodels within ELM and run them independently. And so, for example, we can look at just the canopy fluxes or the part of the model that is predicting GPP and try to break down the model problems down into manageable problems. So each of these kind of subunits will have its own set of uncertain parameters that we're interested in, and in some cases makes predictions of variables that we measure. And so we can constrain functional units maybe one at a time and then bring them back together, combine the information to make predictions with the full ELM. And so we're developing surrogates for parts of these individual process submodels, as well as looking at the full ELM and potentially also doing in the future hierarchical calibrations. Also, something that might be useful is enabling ecological forecasting, so we can take information, observations, calibrate the model, and then make more kind of short-term predictions that might be able to inform field campaigns, for example. Ultimately, one of the goals is coupled uncertainty quantification. So that's obviously a very hard problem. So we're not looking at one component anymore. We'll be looking at many, combinations of components where coupling is important. And so we may have cases where our offline component might be quite well tuned and perform quite well. And so, for example, on the upper left, you can see our offline ELM runs evaluated in ILAM and looking at the bias and latent heat flux. And then we run that same model in the coupled mode, we see very different patterns of bias. For example, the plot on the lower left there. And so biases may be related to other components or the coupling between the components. And so There's obviously going to be a very large computational demand for running coupled models, ensembles of coupled models, and so we want to be able to do this with as small of a number of ensemble members as we possibly can, and apply machine learning approaches where possible, to make the surrogate modeling more efficient, to get away with a smaller number of ensembles. We can also do things like looking at single column models, and so that provides a nice test bed where we can look at land atmosphere coupling for individual components of individual grid cells much more cheaply than we could with a global simulation. So to wrap it up, we talked a lot about forward uncertainty quantification or uncertainty propagation. I think surrogate modeling really is the key to moving forward for these single point, single grid cell type simulations. We've developed pretty well developed workflows, and we're definitely willing to work more with interested folks that might be interested in applying these approaches. And with a combination of approaches, we can get very high accuracy of our surrogate models, and with global relevance, which means not just for single grid cells or single points in time, but looking at these very high dimensional spatio-temporal fields using relatively small numbers of simulations. Again, we've showed a lot of examples for sensitivity analysis or variance decomposition, for understanding which parameters are driving uncertainties, and potentially for reducing the dimension of eventual calibration problems. The global work I showed was primarily with satellite phenology, but we're very interested in extending that to the full BGC version of ELM, which is about 10 to 20 times more expensive when we're considering the spin up, and obviously introduces more uncertain parameters as well. So we think this is possible to do, but we definitely will need to look at the computational resources that are needed to extend this work to full BGC. We're making progress on inverse UQ, so model calibration with Markov chain Monte Carlo is the primary methodology, and the better we can make our surrogate models, the more accurate they can be, the more feasible doing calibration is from site to kind of global scales. And so some of the key advances, I think, are getting these reasonably accurate, spatially explicit surrogates, and potentially also incorporating structural error. So finally, yeah, I think this is a way to enable what we call the MODEX loop. And so bringing in data from observational networks and experiments to directly constrain model parameter and prediction uncertainty. And hopefully this will help us to understand where and what types of observations in the future might be kind of optimal for reducing prediction uncertainty. So I'll stop there. Thank you very much.